Number 10, patient-specific research to enable efficient drug development. As the range of therapeutic options for treating patients with cancer continues to expand, clinical researchers are faced with the enormous challenge of matching the right treatment to the right patient. In order to meet that challenge, novel research strategies that allow individualized testing to predict treatment efficacy will play an increasingly critical role in years to come. This includes interrogation of individual patients' tumors in a way that permits real-time testing for susceptibility to drug or immune therapies. To address the heterogeneity and complexity of treatment response, clinicians will also use computational modeling of tumor-derived parameters, including molecular genetic and proteomic data, tumor cell functional assays, drug dosing, and prior treatment data. Research to confirm the clinical utility of these new testing methods will employ large collaborative clinical research networks, which will provide the number of patients needed to address the diversity of biology and treatment response that are found in cancers that were once considered a single disease with a single treatment.
Thank you very much. And our expert here is Dr. Jeff Engelman, who is the Director of Molecular Therapeutics and Thoracic Oncology at MGH and a leading authority in studying patient-derived materials. And the questions for Jeff are, from the time of diagnosis, how quickly do you have to get this kind of information to be able to impact the treatment of a patient? And then the second question, if you will, is how do you ever control for such N of 1 type studies?
Thanks, Daniel. So I think that the technology that we're talking about is really taking a patient's biopsy, and we already get genetic analyses on these biopsies, and can we do functional studies to find out if they would be susceptible to a specific therapy, a targeted therapy, an immunotherapy. One of the exciting things about these approaches is that you can actually keep the immune cells and the microenvironment with the tumor cells all together to do this functional testing. So it really extends itself to immunotherapy as well. And I think Daniel's hit upon the key challenge is actually getting results quickly enough that would actually impact a patient's care. We've been trying to develop this at MGH. It's not something that we offer patients, it's purely research. And we've assigned a 3-week turnaround as a goal, because 2 weeks seems unrealistic and 4 weeks is ridiculous. So 3 weeks seems perfect. But it does have to be in that 3 to 4 week period time, because patients with metastatic cancer can't really sit around and wait for treatment decisions to go forward. I think the first place this will happen is when patients are on clinical trials where there's like 4 or 5 arms on the trial, and there's no biomarker to direct you onto which arm to go on. And you can see a functional test being part of that. And so I think it will get rolled out slowly into very defined treatment choices, and that the ones that your tumor cells are most susceptible to would be the treatment that you go on. And to see if it has that type of predictive ability, I think on those small scales will be necessary to roll it out in a more broad sense.